Hello, and welcome to our continuing talk on autofluorescence. Today we'll be covering characterizing cellular autofluorescence. Our speaker today is Kate Pilkington. Kate is a cytometry application specialist with Cytech Biosciences, supporting Australia and New Zealand in spectral cytometry and pursuing her passion and promotion of best practices for scientific excellence in cytometry. She has extensive cytometry experience, both Cytech and her previous roles as cytometry facility manager at the University of South Australia in SA pathology, affording her the opportunity to collaborate over a wide range of projects with a strong focus on cancer biology, transplant immunology, and cell trafficking studies. Kate discovered her passion for cytometry during her postgraduate studies at Adelaide University and hasn't looked back. She is also a long-standing member of both ISAC and ACS, chaired the ACS 2018 meeting in Adelaide, and founded the Adelaide Cytometry User Group, Cytomad, to further her mission for cytometry education of local scientists. This session is being recorded, and if you have any questions during this talk, please enter your questions in the question window chat, and we'll be answering them during the talk, and we'll have time at the end for additional questions. I'll now turn it over to our speaker, Kate.
Thank you for the introduction, and thank you for joining us online today. I am really excited to be able to discuss with you autofluorescence characterization using our full spectrum profiling technology. We're going to begin today by learning how to understand the full spectrum profile autofluorescence signatures in our cells and tissues. Then we're going to learn how to visualize the heterogeneity within our samples and how that varies with different laser configurations. And then finally, we're going to go through a couple of case studies outlining how to determine what is the best unmixing option for your sample, and also how autofluorescence can be used in your analysis and how to incorporate additional autofluorescent tags into your analysis strategy. So let's begin by understanding the common characteristics of autofluorescence signatures. And in order to orient you to the characteristics of autofluorescence that we're going to discuss today, let me talk about how our full spectrum profile signatures are created. The full spectrum profile of cellular autofluorescence, just like the signature of fluorochromes, consists of light that is captured from each of the different detectors associated with each of the laser modules on our Aurora and Northern Lights instruments. The signal from each of these detectors is then stitched together to form a fluorochrome signature. There are a number of cellular metabolites and structural components present within cells that are going to contribute to the autofluorescent signatures that we observe. Primarily excited by the ultraviolet and violet lasers, tryptophan, collagen, tyrosine, fibrillin, and indoleamines are major contributors to the distinctive UV peak and that violet hill-shaped emission curve we see. Elastin, NADH, pyridoxine, and folic acid contribute to the emission we see not only from the blue, but also from those violet and ultraviolet lasers. And one of the reasons that we observe increased autofluorescence in dying cells is in fact due to the increase in production of metabolites such as tryptophan, which is going to support the increased energy usage and NADH processing that the cells are going to employ as a last-ditch effort in order to try and survive whatever has triggered them down their death pathway. On the yellow-green laser module, the components that contribute to the autofluorescent signatures include flavins, lipofuscin, and retinol, while riboflavin and porphyrins are the main contributors to autofluorescence that we see in our red laser module. When we think about autofluorescence in terms of our samples, there are three main scenarios we encounter when we're examining our autofluorescence. Firstly, we have homogeneous samples, where all the cells present have a really similar spectrum, and the intensity of that spectrum is similar for all our events. This is the pattern that we're going to observe if we're looking at cell lines or if we're looking at lymphocytes only within a whole blood sample. Please note the distinct UV peak that we're observing, the broad emission curve we see in the violet, and some emission in those early blue channels. But in general, that spectral plot has a narrow profile. We have a narrow heat map in each channel. Then there are heterogeneous samples where our different cells present within the sample are displaying similar spectra, but that those spectra vary in terms of their fluorescence intensity. And this is what we see when we are gating on all cells in a whole blood sample. In fact, lymphocytes, monocytes, and granulocytes display very similar spectra, and they simply vary in the intensity of that spectra. Note the addition of the higher autofluorescence observed in the yellow-green laser, which is characteristic of granular cells, particularly eosinophils, which are going to be rich in flavins. And when we compare this profile intensity with the autofluorescence of lymphocytes alone, we observe a general increase in the median intensity of fluorescence across the whole sample, as well as an increase in the width of that heat map. The spectral plot has more width to it, and we have more outlying events observed in that blue range. And finally, we come to heterogeneous samples where the different cells present within that sample are going to display different spectra and also have variable fluorescence intensity, which is what we would observe commonly in our tissue digests. This digest is from a mouse lung, and note the very intense autofluorescence that we can see, particularly in the ultraviolet channels. Tissue digest samples have a lot of collagen, they have a lot of elastin and fibrin, and these contribute to that very high autofluorescence in this range. Also note a broad range of intensity as denoted by the wide spectral plot, and lots of blue in our heat map away from the median. Any spectral plot that has wide blue bands on that heat map is showing us heterogeneity with a wide range from the median to the outlying events. The previous sample was a mouse lung, and this sample we're looking at now is a mouse bronchoalveolar lavage. The signature heterogeneity is observed across most of the spectrum. Note also an increase in the red autofluorescence, which is from porphyrins in the macrophages that are present in that BAL sample. The spectral plot is relatively broad, and in some detectors we can in fact clearly see more than one signature is emerging. Here we're looking at a mouse skin with very heterogeneous and high autofluorescence, particularly in the ultraviolet and the violet and the blue. And as the sample was for a model where eosinophils were recruited to the skin, we also see an increase in the yellow-green emission. And just like the previous sample, we observe a very broad spectral plot and a number of signatures emerging within certain detectors. In this mouse liver sample, we see very high autofluorescence across the ultraviolet, violet, blue, and yellow-green lasers. And we note a very wide range in intensity from that spectrum. If we were to start adjusting our scatter gate, we can start to understand a little bit more about where these different autofluorescent characteristics are coming from within our sample. When we place a scatter gate around events with low forward and side scatter, we see a medium level intensity of autofluorescence, and this autofluorescence looks a lot like what we observe when we're looking at lymphocytes. But there is still some heterogeneity observed here, as denoted by the wide spectral plot. So this is the first indication that we see on our well-resolved scatter plot that perhaps scatter alone is insufficient to isolate different autofluorescent populations. However, when we move the scatter gate to place it around this group of cells with higher forward and side scatter, we see a very large increase in the intensity of the autofluorescence, and within that group, the signature is relatively narrow and discrete. So far, we have spent quite a while talking about the characteristics of autofluorescence in different tissue types. Now I want to help you find your own autofluorescence heterogeneity within your tissues, what you're going to look for, where you're going to look for it, and how these patterns change when you have different instrument configurations. Here we have those 4 same samples we just examined. We've stacked them atop one another so that you can see where the autofluorescence characteristics are similar and where they differ. The common channels where most heterogeneity is observed are the mid-UV channels, corresponding to wavelengths of light from approximately 450 to 600 nanometers, the mid-violet channels from 475 to 600 nanometers, the early blue channels from 500 to 600 nanometers, the early yellow-green channels from 570 to 650 nanometers, and finally the early red channels from 660 to 700 nanometers. In order for you to visualize the heterogeneity within your samples, you can view the n by 1 permutations of the data from all of the raw channels on your instrument. Now, we have done a lot of digging around in the raw data from many different highly autofluorescent tissues, and as just described, we've made some recommendations to you about where you might expect to find heterogeneity. But for the curious, here's a brief video that shows all 64 permutations on a 5-laser aurora from a tissue digest sample. So rather than displaying all 64 detectors versus one another in an enormous n by n array, we use what we call an n by 1 array. And in this, we plot the first parameter against every other parameter in the data. Therefore, our first plot starts with ultraviolet 1 versus ultraviolet 1. The second plot is ultraviolet 1 versus ultraviolet 2. The third plot is ultraviolet 1 versus ultraviolet 3, and so on until we reach the last plot, which is ultraviolet 1 versus red 8. As the movie progresses, we are simply changing the parameter that is displayed on the x-axis of all the plots from ultraviolet 1, then to ultraviolet 2, then ultraviolet 3, and so on to iterate through all the detectors on the system from ultraviolet 1 through to red 8. So pay particular attention to that top row where we see the UV detectors from ultraviolet 1 to ultraviolet 12. The ultraviolet detectors are often the most informative when we're trying to find distinctive population clusters, and this sample is absolutely no exception. Notice how little differentiation is observed in comparison through those ladder detectors in each laser module. This is an N/1 array of mouse skin sample that does not show much population separation. This array shows violet 16 on the x-axis against all the other channels on the y-axis. The majority of plots show a diagonal streak from which we cannot distinguish unique cell clusters. Only those plots in the first row where the UV detectors are shown are providing any cluster separation, and therefore let's now look at an N/1 array from the ultraviolet 8 channel. On this one, we observe the array, we are able to see a number of plots where multiple different clusters can be identified, and this is an example of good separation. Here is another mouse skin isolation, but this time the N/1 array with the greatest variation for us is plotted using ultraviolet 1. The autofluorescence characteristics of this sample are different from the previous sample because of the different tissue digestion protocols that were employed by the researchers. And it's important to note that your incubation times, your sample source, and enzymatic digestion process can all affect your cellular autofluorescence. In closer detail for these samples, along the top row, here I'm showing you the first mouse skin preparation, and on the bottom row, the second mouse skin preparation. In the case of preparation 1, the best population separation we can observe is on ultraviolet 8 versus yellow-green 1 plot. However, this is a plot that shows very little population separation for the second sample. On the other side of the screen, the ultraviolet 1 versus yellow-green 6 plot really doesn't show much in the way of population separation for our first skin preparation. But when we're looking at our second skin preparation, this plot is the one that gives us the greatest separation of clusters. Now we move to a 4-laser configuration where we do not have an ultraviolet laser. Here we rely on the violet laser to show us the greatest heterogeneity within this sample. And in this mouse liver prep, we see some population separation, but we also observe many more plots that have an arc of population distribution, which really does not allow us to cleanly delineate different populations. And finally, when we are working with a 3-laser configuration, we still rely on the violet laser for providing us with informative separation of populations. In this mouse bone marrow sample, 2 very distinctive populations can be observed. This may not always be the case on a 3-laser system and will be dependent, as always, on your sample and your sample handling processes. So my recommendation for anyone who's interested in knowing more about their autofluorescence characteristics, go find an unstained cells file from your favorite sample and start digging around in that raw data and see what you can observe. If you would like a copy of my raw autofluorescence N/1 templates for your instrument, please email me with your information about your instrument configuration and I can forward you the specific template. Now we have covered what the common characteristics of autofluorescence are, let's have a look at some case studies where autofluorescent properties of the sample have implications in how we analyze the sample and what we do when we're unmixing. When we're unmixing, we have 3 choices. We can do standard unmixing, which is something best suited to samples with low autofluorescence. We can unmix with autofluorescence extraction, which is recommended for samples with high autofluorescence, or we can incorporate additional autofluorescence fluorochrome tags into our assay to characterize and isolate different populations based on our autofluorescence. So let's begin by discussing some relatively common sample types. We're going to talk about lymph nodes, which are quite homogeneous, and splenocytes, which display a small amount of heterogeneity. When we look at the autofluorescence characteristics of lymph nodes, they have a very low autofluorescence, and by now we recognize this as a profile that's synonymous with lymphocytes. When assessing spleen samples, the heterogeneity of the sample is going to be dependent upon what cells we're going to analyze within that tissue. If we intend to analyze multiple cell types within the spleen, for example, lymphocytes and splenic macrophages, then we include a population of cells known for their high autofluorescence, and we also introduce heterogeneity. While we can still see the dominant spectra from the abundant lymphocytes in the spleen, we are also seeing a number of cells that display a significantly brighter and different spectrum as outlying events. If we intend to look at just lymphocytes alone, we can really reduce the scope of the scattergate and in fact observe a relatively homogeneous autofluorescence signature that's characteristic of our lymphocytes. Therefore, when we're planning how to unmix and analyze our experiment, we need to consider very carefully which cells are of interest to us. If we were looking at the heterogeneous sample from the previous slide, we might wish to observe the n/1 permutations within the data and characterize the unique signature from those macrophages and incorporate that into our assay. But fortunately, in the example I'm going to describe to you now, we're interested only in the T cells within the spleen, and therefore the autofluorescent characteristics of the sample are both homogeneous and also quite dim as autofluorescent signatures go. Before we get into the nitty-gritty, remember this assay also looks at lymph node samples. While the autofluorescent signatures of lymph node and spleen are very similar, they are not identical. Therefore, it is important that we record separate unstained controls for each tissue or treatment or stimulation condition in our experiment. And of course, be sure to record as many cells as we can. I like to record as many unstained cells as I record in my multicolor sample. SpectraFlow, the software on our instruments, allows you to define multiple group-specific unstained cell controls for different tissue and sample types, and that allows you to accurately extract the autofluorescence related to the sample in question. Use the group-specific unstained control feature to correctly assign the appropriate control to each sample type. Then, when unmixing, you can choose how you want to unmix. If you do not check the autofluorescence as a fluorescent tag checkbox, you will do standard unmixing. However, if you wish to do autofluorescence extraction, then you need to check that box. Before we begin looking at the data from this case study, first let me tell you a little bit about the panel in use. This is a 24-color lymphocyte panel, and as I mentioned, it was used to examine lymph node and spleen samples. The fluorochromes within this assay that overlap the most with the autofluorescence are found within this region, and in particular the fluor that we observe the most profound effect on is BUV496. When acquiring the controls and samples, all of the single-stained reference controls were on cells. We were able to obtain an accurate fluorochrome signature from every control, and all of the controls were as bright as or brighter than the samples within our experiment. So in order to determine for us whether or not the data is better with or without autofluorescence extraction, we unmix using both methods and then assess and compare the outcomes. When we are assessing our unmixing, how do we tell which unmixing is most accurate? The easiest way for us to assess our unmixing results is by looking at our unstained cells and observing the n/n permutations of all the fluors in the assay. When we're doing this, we are looking for well-shaped populations that are clean and round as much as possible. And unmixing error is going to be observed as nodes that are going to emerge from the negative of that population. Here I'm showing the BUV496 permutation from the unstained spleen sample when we unmixed with autofluorescence extraction. Note the marked protrusion below the negative in all of these plots. When we move beyond the unstained sample and look at our stained sample, this aberrant negative population remains, and when we gate on these cells and look at them on a scatter plot, we see that they are all found in the bottom left corner of the plot near where we expect to find debris. When we backgate these cells, we see that the aberrant negative population tracks as the negative across all the plots. And this population represents approximately 0.5% of the sample, and based on its scatter profile is very likely to be cells that are dying during the sample staining procedure. Ordinarily, when we are staining with a fixable viability dye, we stain at the beginning of the assay. And this means that any cells that die during the assay will be negative for that viability dye, and as a result, they're not going to be excluded by the viability gate, which I've already applied to this data set. When we choose standard unmixing instead of unmixing with autofluorescence extraction, the negative population looks neat and round, and we simply see a little autofluorescence spread into the BUV 496 positive channel instead. In this panel, the antigen conjugated to the BUV 496 is CD45.1. This is a very bright and well-resolved antigen, and if we had chosen a less abundant antigen, for example, CD25, the spread from the negative into BUV 496 might compromise our ability to resolve that BUV 496 positivity. Now let's look at the multicolor panel unmixing results. As a reminder, this is the unmixing outcome when we choose autofluorescence extraction, and when we choose standard unmixing, that aberrant negative disappears, and we observe the expected population distribution for all the markers in the assay. At the end of the day, what we learn from this case is that it is important to assess autofluorescence extraction on a case-by-case basis. The brightness of the autofluorescence and the fluorochromes that you use in your assay are going to dictate whether or not that autofluorescence extraction is beneficial. And in addition, the antigens that are assigned to the fluors that overlap with autofluorescence are going to be of importance. When you're working with very low autofluorescence samples, such as this spleen sample, the spread of the negative introduced by the ordinary least squares unmixing algorithm when extracting autofluorescence may abrogate any gain that you observe by reduction of the negative medium fluorescence intensity. So take the time to assess the unstained and the multicolor samples you've recorded when unmixed and determine the best unmixing strategy for you. Moving away from the question of whether or not to do standard or autofluorescent extraction unmixing, we are now going to examine a case study in which we are working with a complex tissue type that contains multiple autofluorescent signatures of varying fluorescent intensities. Once we have identified and characterized these signatures, we can then incorporate them into our assay as additional fluorochromes, which enable us to exclude cells based on autofluorescence expression, or even incorporate these autofluorescent fluorochromes into our analysis to examine co-expression with other markers of interest. To begin with, let's take a look at this 18 color panel in use with this sample. These are the fluors that are commonly impacted by autofluorescence, and the fluors in this panel that are most impacted by that autofluorescence are AmCyan, BV570, and FITC. The 18 color panel is used to analyze mouse ear skin digests and assess populations of lymphocytes, dendritic cells, and eosinophils. The assay involves looking at wild type and knockout mice and observing the infiltrating immune cells in that ear skin tissue. When all the reference controls were optimized, we still observed a significant unmixing error, and the heterogeneity of this unstained sample is the driving force behind this unmixing error. The reason being that the unmixing algorithm has not been properly instructed about all the fluors present in the assay when the unstained sample cannot have a representative signature derived from a scatter gate alone. Like before, we investigate the unstained sample to determine which unmixing choice is best. When comparing unmixing with and without autofluorescence extraction, high autofluorescence samples will be significantly improved by unmixing with autofluorescence extraction. And in this case, we have unmixed with autofluorescence extraction enabled, and we're observing that AF tag versus every other fluor in the assay. Here you can see the significant variation in the autofluorescence fluorochrome, and we are not observing nice rounded negatives. If the sample is heterogeneous like this, we want to refine our assay and we want to include extra fluorescent tags to represent that heterogeneous autofluorescence. What that error means to us in our multicolor sample is unmixing error from the AF fluor into other parameters, resulting in unexpected populations in several parts of our panel. In this slide, we observe a negative arc into FITC, which results in an indistinct negative population when looking at CD49B versus CD11B, so that we cannot gate our populations correctly. The way that we correct for this is by adding assay-specific autofluorescent tags using our Discover, Distinguish, Designate workflow. So let me walk you through this workflow now. First, when it comes to acquisition of your samples, your unstained control is paramount. we need to identify subpopulations within that whole sample to characterize the autofluorescence from those cells. And some of those cell types are going to be rare. So record lots of events. The more, the better. Personally, as I said, I like to record as many unstained cell events as I record in my multicolor sample. And while we're talking about it, I'm going to remind you to record separate unstained controls for each tissue and record separate unstained controls for each treatment and/or stimulation condition. And this is going to allow us to have sufficient events to identify and extract the unique signatures within that sample in order to add them as new assay-specific tags. As far as the unmixing algorithm is concerned, any autofluorescent tag in your assay is just another fluorochrome, one for which we need to provide good controls that meet all the rules of a good reference control, just like all your other single stain reference controls. To recap these rules, the reference control must be as bright as the expression in the population of interest. The full spectrum of the reference control needs to match the multicolor sample. We need to obtain enough positive and negative particles. And then we come to the fourth rule that states that the positive and negative particles must have identical autofluorescence. And here is where we need to break the mold. We need to define this rule because now we are defining the signature of the autofluorescence as our positive, and therefore we need to identify and use the lowest autofluorescence present within the sample, and this now becomes our negative. To begin the workflow, first we need to discover where the unstained sample shows the greatest separation of populations in our raw n/1 permutations. In this sample, the plot chosen is UV8 versus yellow-green 6. Once we have identified the plot that allows us to visualize those separate clusters, we place a gate around each of these clusters. If we then right-click on the gate, we can export the gated events as a new FCS file. So do this for every single cluster you define. Once we've identified our unique autofluorescence clusters, it is then time for us to go looking for our dimmest autofluorescent population. The algorithm is looking for each fluorescent tag to have a matched negative. When these are matched, the autofluorescence of the negative is subtracted from the positive in order to identify the genuine emission characteristics of that fluorescent tag. Oftentimes you will find that the dimmest autofluorescent signature can be found hiding in the corner with your debris. While we are not interested in the debris, we are interested in extracting a nice dim signature that will act as a negative that will not subtract too much fluorescence from our autofluorescent populations. Now we have our collection of files, including the new autofluorescent fluors and the AF dim. We need to instruct the software about how these tags are going to be used for the unmixing algorithm. So look at the signature of each of your new populations and determine what is the peak channel. In this example, the peak channel is V7, which equals 542 nanometers. Armed with this information, we can now go ahead and create a custom fluorochrome in our fluorochrome library, and this fluorochrome can then be added into our experiment to be included for unmixing. To designate the new fluors, navigate to the fluorochrome tags in the SpectraFlow library. Personally, I like to add a new group to the fluorescent tags group list, as this allows me to bundle all the tags from one experiment into a single group that are easy to identify and incorporate into the experiment. In this group, you can add as many tags as you have identified. And here I'm adding the P9 AF tag, which was maximally excited by the violet laser with a peak emission of 542 nanometers. Now we've defined these new tags, we can go ahead and incorporate them into our experiment. So click on Edit Experiment, find your new AF tags, and then add these fluors to your assay. To match our shiny new AF tags, we also need to add our AF dim sample for the match negative. Do this by checking the box to define additional negative controls for spillover calculation, and then adding a new control called AF dim, which will be associated with cell type controls. Now that you've added the new fluorochrome tags to your experiment, it is time to update your reference control list to incorporate these new tags into your reference control group. Add the new tags, being sure to assign the AF dim as the negative control for each of them. Then the final step in this workflow is to import the FCS files that you generated earlier by exporting events and import them into the corresponding reference control positions, and then go ahead and unmix your assay. So now that I've shown you this example with a fantastically diverse tissue type, let's apply this workflow to our mouse ear skin and compare our unmixing with and without our additional tags. When looking at our mouse skin sample, we observe the raw channel permutations to look for a combination that will best separate the sample into distinct populations. And there are a number of plots that could have been chosen in this array. Each of us has our own preference about how we observe and how we gate data. So the particular combination that I worked with in this data was UV8 versus yellow-green 1. The UV channels are related to autofluorescence from many cellular metabolites that are involved in survival, and the yellow-green 1 channel coincides with expected autofluorescence from flavins, which are a significant contributor to autofluorescence in eosinophils. In this way, I'm really hoping to be able to characterize the autofluorescence from infiltrating eosinophils, which is one of the populations of interest in this assay, as distinct from the lymphocytes, dendritic cells, and the resident skin cells. Looking in detail at our UV8 versus yellow-green 1 plot, we find that there are at least 2, possibly 3 populations that we can gate. The first population is the most abundant population present within the sample. It has a low scatter profile, and the autofluorescence peaks in V7 at 542 nanometers, and we have called it V7 plus low AF. The second population, this kind of extension streak that moves away from the dominant cluster, shows an increase in both UV8 and yellow-green 1 fluorescence intensity. And when we look at these cells, we find that they also peak at 542 nanometers in V7, and we see a much higher intensity signature, and the cells display quite a wide variety of scatter properties. We have called this second population yellow-green 1 plus UV8 plus AF. Our third and final population in this workflow is a population with a marked increase in ultraviolet 8 fluorescence. Upon closer inspection, we find that these cells also have a low scatter profile. They're quite high intensity autofluorescence, but where the cells in the first 2 clusters peak in V7, these cells actually peak at 514 nanometers in ultraviolet 7. And by this one characteristic alone, I know that these cells will be very unique in their signature characteristics. So we have called this population UV8 plus AF. Not to be forgotten is our mission to find some very dim autofluorescent cells. In this sample, I have found this little box returned cells that display a very dim autofluorescent signature, which has very little heterogeneity in it. When I gate on the lowest intensity cells from the peak channel of violet 7, I export these as my new AF dim population. And then when we are unmixing, we need to look for this tiny little blip on our scatter plot to gate around them to find these cells again. On this graph, I'm now showing you the normalized spectra for each of the autofluorescent tags we just identified for this skin sample. Just like other fluorochromes in our assay, the determination of whether or not that autofluorescent tag is compatible for unmixing in the assay is going to be dependent on the similarity index of those fluorochromes. Fluors with a similarity index below 0.98 can be useful for unmixing together. The easiest way for us to go ahead and assess the similarity of the AF tags is in fact to create a new experiment with only those tags in it. Then we use the QC tools within the unmixing wizard to evaluate the similarity of these autofluorescent fluorochromes. Here we see that 2 of the tags are unique and 1 pair shows 99% similarity. So armed with this information, we can make a decision about which of these tags is likely to be most useful for incorporating into our unmixing workflow. So let me walk you through my decision tree for choosing unique autofluorescent tags. I always start by identifying the most abundant population. This is going to represent the majority of cells within the sample. In this sample, the most abundant population is that V7 plus autofluorescence population. Therefore, we definitely want to keep this tag. The next most abundant population is the UV8 positive AF tag, which is 75% similar to the V7 population. Therefore, these signatures are unique, and we're going to keep this tag. The final and least abundant population is the YG1 plus UV8 plus AF population. This does not have a unique signature. It is 99% similar to the signature that we found in that dominant V7 population. Therefore, I will not be incorporating this tag into my experiment. So when thinking about the new AF tags that we're adding, it is also important to consider how these fluorochromes are going to overlap with the other fluors that are present in our assay. In this assay, AmCyan and BV570 in particular are really highly overlapping with the autofluorescence of the cells, and this will impact our ability to resolve the positive events from the negative as a result of the spread of the negative population. In addition, the two autofluorescent fluors themselves are also highly overlapping with one another. When the new fluors are incorporated into our assay and we unmix, we can assess the similarity and complexity indices of our upgraded panel, which has been increased from 18 colors to 20 colors. As predicted from the normalized spectra plots that we just observed, the two autofluorescent tags show the highest similarity between one another and AmCyan and BV570. And as always, the place we look for guidance on understanding if our unmixing choice is a good one is by looking at the autofluorescent tag versus every other fluor in the unstained sample. Again, as we saw before, here we see considerable sample heterogeneity and significant unmixing error when we do autofluorescence extraction without any additional AF tags. But when we incorporate the tags examined a moment ago, we see much improved unmixing, as the unmixing algorithm has now been instructed about the variable AF signatures within the sample by us providing additional reference controls from the unique portions of that unstained sample. When it comes to our analysis with these new tags, the new AF tags can be used for analysis in conjunction with other fluorochromes in an assay such that the antigen co-expression with the discrete AF population can be determined. Note where the UV8 medium intensity cells co-express CD90.2, or where the UV8 bright cells co-express CD11b, and that the IL4 expression is limited exclusively to those UV8 high autofluorescence cells. To show you a side-by-side comparison, on the left here is the 18 color panel, unmixed with autofluorescence extraction and no additional tags. Note the indistinct negative population between CD49b and CD11b negative cells, the slight protrusion of the CD49b negative events into the positive, and the minor bulge at the top right of that CD49b Siglec-F negative cell population. When we look at the same data after we've added the two new AF tags in our 20 color panel, each of these errors is now resolved. Note the much tighter negative populations, and these comparable data are scaled identically, so the impact of the improvement is hidden within the compressed scaling of that negative. So when I optimize the scaling of that 20 color data, now we can see that our negative populations are clean and well resolved, and in particular, we can determine where the CD11b and CD49b double negative events begin and end. In this presentation, we have learned a lot about what cellular autofluorescence looks like, how it changes, and how we can view it and understand the subtleties within our samples. So there is a whole spectrum of information out there in your data, and it is just waiting for you to discover it. If you are thinking about embarking on a new panel and you are working with autofluorescent tissues, I really recommend that you go ahead and analyze your unstained sample prior to designing your panel. Because if you know your autofluorescent characteristics and from roughly which cells these different characteristics emerge, you can now make much more informed decisions in your panel design. Do you have some unstained samples sitting in your data archive? Why not go check them out and start diving into the wealth of free information that is already buried within your data set. I no longer think of autofluorescence as just a challenging background component that I need to work around. With full spectrum profiling, we're proposing a model where you can incorporate that autofluorescent characteristic of the cells in your assay, obtaining information from your sample with a label-free analysis component. Now that I've spent a lot of time working with autofluorescent samples, I find these the most interesting samples to analyze. But never forget that now your unstained control is arguably your most important control. Be sure to record lots of events. Don't just throw a few spare cells in this tube. Treat it with the care and consideration that you treat your multicolor sample. Then, when you are ready to understand what is going on with your unstained samples, observe those n by 1 arrays of the raw data to find your autofluorescence heterogeneity. And finally, observe the n by 1 plots of your unmixed data to assess the best unmixing strategy. I'd like to thank my colleagues and fellow researchers who've contributed to this webinar. Many thanks to Maria, Diana, Yassine, Juliet, and Elizabeth. These are a group of colleagues who have been instrumental in developing our understanding of autofluorescence heterogeneity and how we communicate this information with you. Next, I'd really like to thank Laura Ferrafont from the Malagan Institute of Medical Research. The data presented in case study 2 was from Laura and one of her users at her flow call. And finally, Ray Steptoe and David Sester from the Translational Research Institute of Australia and the University of Queensland Diamantina Institute, whose data was presented in our first case study. We have another webinar coming in our series of webinars about autofluorescence. It's not over yet. If you're interested in learning more about considerations of autofluorescence when designing your panel and the use of autofluorescence as a label-free method in some further case studies, do scan that QR code and sign up to our newsletter to be notified when Yassine will be presenting the next webinar in the series. Thank you so much for joining me for this webinar.
Great. Well, thank you very much, Kate. Thank you all. That is all we have time for today. And we really appreciate you taking the time to come and listen to us talk about spectral cytometry. And with that, I'd like to end this session. Thank you very much,
Thank you so
Kate.
much.